From Microbe TV, this is Immune, episode number 24, recorded on September 17th, 2019. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back. We're at number 24. 2 years.
2 years, that's right. And from Durham, North Carolina, Steph Langel.
Hey there. Happy 2-year anniversary of Immune. Wow. Can you believe we got here?
Goes quickly. Yeah.
It goes by very quickly.
Yeah. And we are almost over September. It's like halfway through.
My goodness.
It's crazy.
I marvel.
Time goes. Mm-hmm. It marches on. How's your weather?
Time,
is your weather at Ithaca? Is it cooling? Is it cooling off?
It's a little cooler, yeah, but it is spectacularly beautiful today. Clear skies. It's a gorgeous fall day.
How about down in Durham?
It's pretty warm. Yesterday, I think it was about 93.
Wow.
Oh my God.
So we're still hanging on down here. Still hanging on. But, it's fine. I'm trying to complain about the weather less, but I think it's one of our favorite pastimes as humans. It's always something to talk about.
You didn't have any storm business from that hurricane a couple of weeks ago, right? What was it called?
Hurricane Dorian. And I was kind of surprised. They canceled school both Thursday and Friday, and we maybe had just a storm, just a mild storm. I mean, I get it because if the roads flood, particularly in rural areas, it's very kids get stuck. But I'm just like, oh, dang, they cancel for anything down here. It's pretty mild.
It snows.
Yeah, but they didn't know if the hurricane was going to turn in, right? Because then it could happen quickly, and then
it could be
a problem.
Real problematic, for sure.
If you like what we do here on Immune and all the other podcasts in the Microbe TV network, consider supporting us financially. Go to microbe.tv/contribute. More on that later. Today it is Steph's turn, and she picked a paper where one of the authors' name is Stephanie.
I know. That's the only reason. I just looked for Stephanie.
I don't believe that.
Yeah, so this paper first popped up on my Twitter, as many good papers do. The title of this paper, I'll say first, then the authors and where they're affiliated. It's called Identification of a T follicular helper cell subset that drives anaphylactic IgE. The first author is Uthaman Gauthaman. The last two authors, I think they are described here as co-corresponding authors, as Adam Williams and Stephanie Eisenbarth. And Stephanie is from Yale, and Adam Williams is from Jackson Labs. And also there's a program with it at UConn, and they work together to identify this particular brand new subset that drives anaphylactic IgE. So I'm going to go through just introducing some of these key players here, particularly ones that I mentioned in the title. So before I start, I'd like to ask either of you, do either of you have any type of allergy?
Nope.
No
allergies. Now,
Isn't that shocking?
It is shocking.
Do you?
Considering the numbers. I do not. Nope, no allergies.
Whoa, I think we beat the statistics there.
I think.
Do you know when I ask this of my immunology class, and usually about 50 to 70% of the students raise their hand. And then the other question I ask is, do you know somebody who does? And then all the hands are up. So right. either you have it or you know somebody who has it and directly affects you.
Yes. And
I always ask my students first day, who has a virus infection? And, 10 people raise their hands and I say, wrong.
Yep.
Wrong.
Every single one.
And it never changes. Of course, from year to year, I have different students. So,
right.
Anyway, so go ahead. But at some point, there are two words in this title that I'll bet are already stumping some listeners.
Yes.
Because they could stump me.
Right? Yes, sure.
And
sure.
One of them is follicular and one of them is anaphylactic.
Yes. Well, before I move on, I was curious, though, do your kids have allergies?
Yes. One.
Okay.
Yeah.
So that's interesting
they do. Mm-hmm.
because that leads me to a statistic that we have been seeing an increase in atopic and allergic diseases, food allergies, environmental allergens and allergies for over the past like 30 to 40 years. And it really is affecting millions of people in the United States. And we might think of allergies as something as not very life-threatening, like, okay, maybe your nose is running, you have itchy skin or itchy eyes, but it really can be a problem, particularly something called anaphylaxis. And I will describe a little bit what that is, but the problem with that is if it reaches this very severe life-threatening form of allergic immune response, it can be problematic. And so, Vincent, would you like me to describe those two things first, or first describe how an allergic response happens?
Yeah, you'll get to them, I'm sure. I just wanted to say to listeners who don't know what they mean that we'll get to it.
Great.
But also, Steph, do we know why allergies are increasing?
There are many theories about that. I don't know if you guys have heard of the hygiene hypothesis.
Oh, yeah.
That seems to be one that pops up quite often, is that because we're not exposed to the same amount of environmental antigens, allergens, because we don't interact with our environments as much, that this leads to this hyperactivity of our immune system to these allergens. I think that there is some validity in that. There is evidence that children that are raised on farms that have a high exposure to allergens on that farm, and particularly ones who grew up with pets, not just one pet, but it has to be multiple pets. So it seems to be that this low-level stimulation over time helps to decrease the rate of allergies. And so that's one theory. There's a couple others.
I think there's some info also about microbiome, right? Because as we change how we're feeding babies from breastfeeding to bottle feeding, now it's going back the other way, but we've shaped changes in our microbiome that also are important for development of the immune system in general. And so it could develop the immune system slightly differently and predispose to allergies as well, right?
Yes. And we, for a long time, and particularly in our children, because they get sick, have been using a lot of antibiotics and may not be using them per the prescription. So there's some evidence that antibiotic-induced allergen sensitization is a problem. So yeah, there are a couple different reasons. I don't think we've pinpointed one, but I think they all work together to really this issue. So I guess I tell you, raise your kids outside more could be the
well, I didn't grow up on a farm, and we didn't have any pets, so of course there are always exceptions, but it's probably not the only thing, right?
Right. Probably not genetics is going to play a role, and how, if you lived in the city, so you maybe had a different range of antigens in your environment. So but definitely the trend would be such that if you weren't raised around some of these things, that can be predisposed to allergies.
So if you look at, say, populations, and I'm sure you can find island populations still where they have a extreme outdoor lifestyle, the houses are thatched and they don't have any screens or AC and they're outside a lot. Are the rates of allergies lower in those populations?
Correct. So they're not seeing the same increase that Western countries are seeing, USA, Europe. There's a lot of other issues. I think helminths and parasitic infections are one of these other mechanisms where if you are infected with a parasite or a helminth pretty much over the course of your life at each stage, that can increase your sensitization as well. So yeah, it's definitely a Western country specific phenomenon.
Okay.
Yep. So what happens during an allergic response? And I'm first going to introduce the first molecule, which is IgE. And the discovery and characterization of this antibody class was really probably the most crucial advance in our understanding of allergies and allergic disorders and how IgE is produced. It's produced just like any other immunoglobulin, an antigen or allergen-specific. IgE requires that the antigens are taken up by dendritic cells or B cells or anything that's an antigen-presenting cell. And in the presence of two key cytokines, interleukin-4 or interleukin-13, provided early in the process, and that can be provided by a bunch of different cell types, a couple of which we'll talk about in a bit. They present that processed antigen to naive T cells, and those T cells acquire something called a T helper type 2 cell phenotype. So there's T helper type 1 and T helper type 2, and T helper type 1 responses are going to induce interferons and help to stimulate an antiviral responses, whereas Th2 cell phenotypes are going to engage B cells through their BCR and other stimulatory molecules for themselves to secrete IL-4 and IL-13, but then undergo class switching as we know it in B cells to produce the IgE, which is defined by the constant region. They switch, we've discussed IgG, IgA, IgE, and that class switching to IgE is really what defines that immunoglobulin isotype. And so by this feedback message, so once an individual has developed IgE antibodies to a certain epitope, there are multiple mechanisms that can lead to a more robust and diverse IgE response. And I'm going to describe two of them. One is a low affinity IgE and a high affinity IgE. Now, the low affinity IgE does not result in this type of acute allergic reaction that leads to anaphylactic shock, as I mentioned earlier, kind of this more life-threatening allergic response. But the high affinity IgE, the central event is the IgE binding to a particular receptor on the surface of something called a mast cell or basophils. And once the IgE binds to this Fc, it's called Fc epsilon receptor. These Fc receptors are on the surface of many type of innate immune cells to provide effector functions. But this high affinity IgE binding is what stimulates the mast cells to degranulate and release mediators that are stored inside these inside these granules in the cytoplasm. And these granules are preformed and they have things inside them, things you may have heard of, like histamine, proteases, cytokines, TNF alpha, which is tumor necrosis factor alpha. And so when they release those things, there's a lot of downstream effects that happen. So you're recruiting more Th2 cells. Dendritic cells are maturing inside a specialized compartment. I'll explain in a bit. And so there's also release, I mentioned, of histamines, which is going to cause things like vasodilation. It can increase your heart rate, vascular permeability. Things like prostaglandins can actually then cause peripheral vasodilation. There's a lot of different mediators that really cause vascular permeability and can lead to kind of organ shutdown if it's not regulated. And
so I have a question. So we take antihistamines, and they work really well on your general runny nose, itchy eye type of allergies, but we don't typically think about them working well on these anaphylactic type of responses, which we might want to actually say what happens when anaphylaxis happens.
Right. So, I mean, anaphylaxis is going to happen when you have, when you really have this vasodilation, you have a drop in blood pressure. And so that response can lead to death because your organs start shutting down. And so it's this systemic inflammatory response that produces that blood pressure loss, organ failure, and is the most severe form of allergy.
And these are the ones where it's usually to something like penicillin or a peanut allergy, or what are some of the other ones? A bee sting.
Yep. Mm-hmm.
So these cause this systemic response.
Yes. Yep. And so that's kind of difference between low affinity and high affinity. How B cells are mediating that is really unclear. Mm-hmm. And so can
I ask you this?
Yeah, sure.
So they're different IgEs, right? Because they've been produced, as you'll probably tell us, in different ways. But the affinity refers to the affinity for the antigen.
It's the so what happens is it's the affinity for IgE to bind these Fc receptors on the mast cells, and it actually is increased. So what the other thing that happens is when you have an increase of antigen in the environment, you're stimulating all these cells to continue to recruit, which is like this positive feedback loop, promoting IL-4 and IL-13 secretion, and you get these things called immune cell complexes. And so when IgE is binding to both the antigen and the Fc receptor on the mast cell, you have cross-linking on the mast cell that really increases the downstream stimulatory signals. And so that happens. I mean, cross-linking can happen in a variety of different ways, but really that's what the high affinity is, what drives that cross-linking, and it just really stimulates mast cells to release an increased amount of mediators.
So the low affinity will not bind to the Fc receptor very well. So you never get this degranulation, right?
Right. So not as well. It seems to be that if you have, one IgE that's bound to maybe one antigen, it seems to be this immune complex that when you then have, and it has a lot to do, I think, with the topology on top of cell membranes, is that when you have a cross-linking where you have two, so if you imagine your antibodies, the constant regions, that's what's binding the Fc receptor on the mast cell. And if there are two binding, and then you have them complex to the same antigen, it brings together the Fc receptors closer. And so having them be closer can help then further activate that cell in a greater way, a higher magnitude.
So are these epsilon Fc receptors only on these mast cells and basophils? Are they widespread? So why is it that this antibody cross-linking leads to degranulation, whereas antibodies on other cell types, they should be cross-linked also by their antigens? It doesn't happen. You don't get anaphylactic responses or allergic responses. Why is that?
Yeah. There's other receptors like CD23 that mediate low affinity IgE binding, and they're on a variety of cell types, not just mast cells. But why the mast cells have a higher affinity for IgE, I think it's because that's a why question. No,
it's not I don't mean to ask you a why question. I mean to ask you, is it correct that only mast cells and basophils have high affinity IgE receptors? And if so, does that explain why only IgE causes these types of reactions and not other?
Correct. So the highest amount of Fc epsilon receptors is going to be found. There are mast cells, basophils, and eosinophils, and I think there's like something in the epidermis, Langel cells. I remember we talked about Langerhans cells, I think, are another one. But those, it is specific to really these cells with preformed granules, whereas other low affinity receptors, they can be found on epithelial cells, B cells, but the high affinity ones are only found on mast cells, basophils, and eosinophils. Okay.
Now, these granules that are released, what's their normal function? They're not there to give us allergic responses,
right? Right. Well, when they're released, you would have to, particularly in helminth infections, you do want to have low levels of release to help clear type 2 Th type 2 related infections like helminth infections. I think that, you do want to have an ability to increase the permeability of your blood vessels to help things like neutrophils get through. Neutrophils then could migrate to tissues to help fight off different types of infections. So I think you would want to have these types of chemicals to help cells move throughout your body, but you do not want too many of them.
Okay.
Or too much release.
Got it.
Yes.
Those mast cell granules also contain things that are like anti-parasitic or antibacterial as well.
Yeah.
So when they degranulate, it's not just to cause you to have an allergic reaction. They do actually have a normal function in the immune system. Whenever I teach hypersensitivities, which encompasses all of these allergic responses, of which the IgE mediated anaphylactic responses are one, I always teach that we learn about the pathologic aspects of that immune response, but there is actually a normal function for that. You brought up an idea earlier about this hygiene hypothesis. Mm-hmm. And if individuals have parasites or they're in a dirty environment, I always think of it like it's kind of occupying the problem teenagers so they have something to do so they don't cause a problem. But when you clean up your environment and you don't have anything for those cells to do, now they start making reactions to things you don't really want them to respond to, which we would normally have our immune responses balanced. But now you start creating these allergic type of immune responses to innocuous antigens, which you shouldn't respond to. That's simplifying it. But generally, you can kind of think of it that way. There are normal functions for all of these responses, but we don't usually notice them. They kind of happen and they're on the low level. But now as we clean up our environment, now we're generating these immune responses that are capable of these life-threatening sequelae because we have these types of IgEs.
One additional clarification. Low affinity IgE, which is produced, in helminth infections, say, it will still lead to some degranulation, but not to the extent that high affinity IgE would. Is that correct?
Right. And then I think the other thing about low affinity IgE and what it does, it kind of helps to block the receptors for when you have high affinity IgE, which is why, when you're sensitized to, like if you have a tolerance and you're able to have antigen specific IgE, but then that doesn't lead to the binding of high affinity, that's kind of that balance you want to strike. I think that, but to get the release of what we're talking about in terms of an anaphylactic response, it would have to be high affinity. Plus, we're going to learn a particular set of cytokines to help drive that response as well.
I think that, so we did a paper on TWIP some time ago where one of the parasite strategies is to exclusively induce low affinity IgE, because with high affinity you get a flushing of the parasite out, right? And they don't want that as much as they want anything, but that they have evolved not to do that. So that's a cool strategy, right?
Yep. Very cool strategy. Yes. Yeah, it's amazing. And I think that, it's not only, IgE is mostly, and we think of helminth infections and allergies, but I think that, there are other roles potentially in terms of like the microbiome and what it's doing in tissues and just its interaction with bacteria and viruses too, that when you think of kind of the microbiome being not just bacteria, but also what parasites are in there. It's interesting to think about.
So, you asked earlier if our kids had, so years ago I brought my daughter to an allergen because she had and they put this thing on your skin.
Yep.
It's like an array with, and I think she had a dozen little needles and she was allergic to every one of them.
Oh, no.
She had a row of 12 dots of red, and the doc was saying, okay, this is grass, this is wheat, this is this. All of them.
Do you know what, though? That's an interesting phenomenon called atopy.
Mm-hmm.
So, this whole idea that once you develop one allergy, that you have that what Steph was talking about, generation of the Th2 type of response. Now you have the cytokines around in higher concentrations that promote the IgE.
Yeah.
And so you tend then to develop additional allergies. So that's another interesting thing that I often ask my class. If they, I ask them, how many of you have allergies? And a certain number of them will raise their hands. I said, how many have 2? And, at least half of them keep their hands up. And I'll ask 3 or 4 or 5. There are sometimes people that are allergic to, like your daughter, many, many, many different things.
Right. Yeah. And so in this paper, one of the main focuses they wanted to understand, and that's not understood, is what determines the regulation of B cells to either produce low affinity or high affinity IgE during an allergen response. And one of the cell types that they talk about is something called a T follicular helper cell. And why they bring that up is because in recent years, they've shown that these TFH cells, which I'll refer to them from now on, are really essential for the development of antigen-specific IgE responses. For a long time, it was just thought that these Th2 cells were the main drivers of IgE, but then these T follicular helper cells have been really, people are diving deeper into the different types of TFH cells, and they have shown a role in IgE, particularly ones that produce IL-4. So we know IL-4 is important in producing IgE, but it wasn't that when they were looking at this certain cell type, the IgE wasn't produced all the time, but TFH cells secreted IL-4 all the time. So trying to decipher and understand that better, and why they really cared about TFH cells and why people are interested in them is they're really integral into development of the germinal center. and they function inside of these structures, these germinal centers that are inside of within secondary lymphoid organs, so lymph nodes in the gut or in other places throughout your body. They form within these B cell zones of the secondary lymphoid organs and during mostly an ongoing immune response. So B cells within these germinal centers, that's where they're undergoing this proliferation, diversification, and allowing the production of many different types of antibodies. And it's these TFH cells that help B cells to expand and to increase their affinity and longevity. So it's really this TFH interaction with B cells in addition to this regulatory role of IgE that got them interested. And so that led them to this hypothesis that does a Th2 important cytokine IL-4 positive TFH cells, do they play a role in the high affinity IgE induction?
Can I ask you, what does it mean to be follicular? Does that mean the cells are only in the lymph nodes, a part of the lymph nodes?
Well, yeah, and it's interesting. So even though these are T follicular helper cells, I mean, they're helping B cells. And so there are zones within the lymph node. There are B cell zones and T cell zones are more in this like circle and more in the center. And then B cell zones are more outside in these like little zones kind of circulating around the T cells. And this type, because they need to help B cells, they're actually in the B cell zone.
So a regular Th2 cell would be where then?
Well, a regular Th2 cell they're also helping B cells. So they do interact and go, like a naive T cell is going to move from the T cell zone and then move to more towards the germinal center. But the TFH cells are always in the B cell zone. So they're more of like a differentiated T cell. And their stimulation is really because of dendritic cells. So dendritic cells that are interacting with the antigen, that are bringing the antigen to the lymph node, I think it's IL-6 they secrete, and that helps differentiate naive CD4 T cells to TFH cells. They migrate then to those B cell zones to interact with B cells.
So frankly, I'd never heard of T follicular helper cells. I've heard of TH1 and Th2 cells, but now we have TFH2. And that is simply because they sit in that zone. They don't move around the lymph node at all, right? I mean, I know there are other differences too, and we'll get into those.
I think they got their name because they're really in the B cell follicle, which is the part of the germinal center where the B cells do all of this class switch.
Okay.
So it's a T cell in that B cell, in the germinal center, in the B cell zone, like Cindy said, yes. And that's what gives them their name. So they're pretty unique. And then their function is the affinity of the antibodies produced by B cells. And so they're really of high interest because if we're talking of a humoral response, trying to generate very specialized antibodies like broadly neutralizing antibodies in HIV and vaccine development, they're really of high interest.
So we also have TFH1 as well as TFH2, and then we have TH1 and TH2 now, right?
Very confusing. Yep.
So we have actually 5 now helper T cells that are involved in antibody generation. TFH1 and 2.
More than that, but
what are the other ones? What are the other ones?
I don't know.
Well,
there's so many different kinds.
They describe in the paper that TFHs, I don't even know how many, but there's like TFH2, TFH6, and we're going to describe a new TFH that was discovered, and they're named based on the function. Cindy, I don't know. Did they use
like TH1 and TH2 were defined, I think, because there were only T helper cells, and then there was a second kind of T helper cell, so they had to call the first one TH1 and the second one TH2. And then they decided, well, this is going to get too complicated, so instead of going TH3, TH4, they started naming them based on either function or cytokine. So we have things like TH17 cells that are important in autoimmune disease, and T regulatory cells. Then the T follicular helper cells, I think the idea is they thought there was one type. Now we know there are multiple different types that make different cytokines that promote different types of B cell isotype switching. So they, instead of trying 1, 2, 3, which they, made no sense, and that's usually how immunologists name things, they figured they would name them based on the cytokines they make, which is how they got to this strange name of the T follicular helper 13, which is also confusing because that's not the only cytokine they make. But
right.
Yeah, there's just no good way to name these things.
Yes, but I think we could think of T follicular helper cells as a very important, but actually relatively rare, especially when you start subdividing them. They're not a common subtype, and they can be found in circulation, but at low levels. They're mostly found in the germinal center, particularly during an active infection or vaccination, because again, their goal is to help B cells class switch, affinity maturate, and they're pretty special. And I think, and we'll learn a little bit more why in this paper, they were special for this antigen-specific IgE. So do we think we have everything defined before
I just want to say that there are only 7 different kinds of viral genomes out there. That's all you need to do everything.
Apparently, we need many more immune cell types to fight all those viruses.
Yes, many cell types. And I think the other thing that makes immunology complicated is as we develop better technologies, we have different ways of identifying immune cell types other than just their surface receptors or the CDs that are on the surface. That now there's transcripts inside, and we can do single cell looking at. So as we increase our ability to define them and define them, as Cindy said, based on function and not just like how we discover them, I think every cell type we discover will have many types and more subtypes, and we'll keep finding new ways to define their function, which is exciting. I think, somebody had once told me in my training that they felt like immunology, everything was already figured out. They know all the cell types, and it's just, they wanted to do something like virology because it's always mutating, and that cells were boring. But I don't think that that's true. There's a lot more to learn.
Well, I think it'll be figured out when we have a nomenclature, like you said, that's functional and not just, based on molecular properties.
Yeah. So I was just looking up something to see if I could find the origin of follicular helper T cell, but I think it's really just because it's in the B cell follicle. But I just read something really funny. So it said that Keith Yamamoto has, he's an immunologist, has said scientists would rather use each other's toothbrushes than use each other's nomenclature.
That's perfect. That's true.
I love it because I really think that, everybody wants to put their own name or stamp on an immune cell because there's so many different ones. And so one of the problems we run into is it a different differentiation state, or is it actually an independent cell type? Or is it a one type that kind of specialized its function to one direction or another? And
it's pretty important.
Yeah. So we started looking at cells and grouping them based on surface antigens. So you have CD4 T cells and CD8 T cells. And now that we have the single cell cloning, now you can go down and compare thousands of genes and what's different from one group of cells to another. And, reality is that you blink at a cell and it's going to be different than the cell next to it, right? So where do you draw the line as what's an independent cell type, which is not? And I think that's sort of where we're all struggling right now to find our way with all this big data.
I agree. So with that hypothesis that IL-4 positive TFH cells play a role in high affinity IgE, they started with using a mouse model that recapitulates a particular disease seen in a gene called DOCK8 deficiency, which I think it was like 2004. It was defined as the monogenic response, whether it was cause of something called hyper IgE syndrome. There's a couple genes that can cause hyper IgE syndrome, but one being DOCK8. And they found that people with DOCK8 deficiency have really bad allergies, food allergies, asthma, and it can cause a lot of issues. I would say that DOCK8 deficiency, even though it causes hyper IgE, it actually has severe consequences for viral infection as well. So CD8 T cell survival and function, dendritic cell function are all altered in this DOCK8 deficiency. But I think what they wanted to capitalize on was the DOCK8 deficiency causing this hyper IgE syndrome. And being able to study molecular mechanisms of IgE induction using DOCK8 deficient mice. And if they could recapitulate the hyper IgE syndrome found in people. And so the first thing they did was they were able to knock out DOCK8. And looking back at some previous literature, Stephanie Eisenbarth, the last author, has worked with this mouse model in the past. That some of the dendritic cell things I'm referencing were from her work. And so the first thing that they were able to do is in these DOCK8 deficient mice was that they did recapitulate this hyper IgE syndrome. And they were able to then continue, and they looked at type 1 immunization. And to do immunization and drive this type 1, they used something, it's called hapten immunology, or basically they took a hapten, which is just a peptide that's too small to elicit an antibody response on its own. And they conjugated it to albumin, which is the hapten's carrier protein. So together, this NP-OVA or ovalbumin can help to stimulate an antibody response, but then we can look at the specific antibodies to, and NP has a longer chemical name, which I didn't write down in my notes. I have to go back to look at the paper, but just that it is a long chemical that by itself cannot induce an antibody response. And with this NP-OVA as a model antigen, I think we just say model antigens because ovalbumin is kind of one of our oldest and best described immunogens. And they paired that with LPS, which LPS stimulates TLR4 and actually drives a type 1 IgG1 based immune response. However, in DOCK8 knockout mice, it actually decreased high affinity IgG1, but then increased high affinity IgE. And so even though, this type 1 immunization is supposed to not be associated with Th2 cells, they knew it was a T cell dependent response, and that these DOCK8 mice, there was something wrong, that they weren't able to control high affinity IgE production. So that helped, as we talked about last month, about developing mouse models that recapitulate disease so they could further investigate why this may be happening. And so they're further wanting to confirm, okay, this DOCK8 knockout mice, it's producing this hyper high affinity IgE, but does that actually cause anaphylaxis? They do something called a passive cutaneous anaphylaxis assay, or PCA assay. And it's a pretty neat assay. I thought what you do is you can quantitate anaphylaxis by using a dye and to measure changes in vascular permeability after an antigen challenge. So what they did was they took serum from mice that were immunized with a high affinity antigen, injected it into the ear of a naive mouse, waited 24 hours, and then challenged that recipient mouse with the same antigen, and then waited like 30 minutes, harvested the ear, and you can see how much vascular leakage is happening by taking their ear fluid, processing it, and kind of measuring, almost like you do measure an ELISA, by the amount of dye that has permeated into the ear. And so what they were able to show was that the DOCK8 deficient mice had an increase in vascular leakage, to help prove that it was high affinity IgE that causes anaphylaxis and not low affinity IgE. And so once they did that, then they wanted to try to further define, what is this inability to regulate the production of IgE by B cells. So they're like, okay, let's look at our cell types. They mentioned first right away that they know T regulatory cells, which we've talked about, play a role in allergen tolerance. So what they did was they knocked out DOCK8 specifically in T regulatory cells, but that deletion didn't change the affinity of IgE responses. It wasn't any different than the controls. So they said it's not T regulatory cells. Well, let's move on. We know about T follicular helper cells. We know about their role in IgE production. And then when they knocked out this DOCK8 specifically in T cells, that they were able to then look at the mice, and they had an increase of T follicular helper cells that produced these two key cytokines, IL-4, but also IL-13, while the wild type T follicular helper cells just expressed IL-4. So I had mentioned before that IL-4 was really key in kind of this low affinity IgE, but when paired with IL-13, potentially this was a way to induce IgE. And this cell type in the DOCK8 deficient mice with hyper IgE had the IL-13. So from then on, they said, well, the unique part of this cell subset is the fact that it produces IL-13. They also showed it produced IL-5, which you would ask, well, why didn't they call it, TFH IL-5? Because it also produces that. And what I kind of gathered from this, as they walked us through their paper, was that the ones that were driving the hyper IgE were always producing IL-13, but only sometimes producing IL-5. And so that's why they named it IL-13, that was kind of the key cytokine that helped define this very special cell subset. And then in addition, they go on to try to further describe this cell type that's expanding in DOCK8 deficiency, that's expanding in correlation with hyper IgE. They mention something, a transcription factor called GATA3, and they mention a transcription factor called BCL6. Now, these two transcription factors are kind of considered master regulators. So a transcription factor that really is regulating the differentiation of this specific cell type, that if you were to knock out this transcription factor, this cell type would not develop. So TFH cells are really defined by BCL6, and Th2 cells are really defined as GATA3. And so they also found that these TFH cells, they express GATA3, but they also express BCL6. And so leading us to think that, well, maybe, and I kept this in my mind as I was working through the paper, we're defining this as a new cell subtype, but they're also expressing transcripts that are specific to Th2 cells. So what does that mean in terms of, is this a cell type that is in transition, or is this its own cell type? And I think we'll continue to learn more about that argument in a bit.
Gosh, just parenthetically.
Yes. I
talked to someone I did a podcast in Denmark, and she worked on this immunodeficiency called monoMAC. Have you heard of that?
I think so, yeah.
Yeah, they have amino acid changes in GATA2.
Oh.
I mean, they were interested in, they have impaired immune responses to herpes viruses, those individuals, and so it's GATA2. Wow.
Wow. Now, it's not GATA3, though, it's GATA2.
Right. Yep. Okay. It's two different
I'm sorry,
ones.
It's two different ones. Yep.
Yeah. I don't know how many different GATAs there are.
Too many.
Too many?
Yeah.
Sorry.
One too many. More than one is one too many.
I was just excited that I knew something.
Well, I think that was still a very important piece of information that we can put in our back pockets for later if we talk about herpes viruses.
Okay.
So, so kind of further trying to define this subset, they also mention IL-21, and they, kind of what IL-21 is something that helps B cell differentiation and is produced by Th2 cells, but the T follicular helper, the TFH13 cells, they actually secrete less IL-21 than when they're DOCK8 deficient compared to controls. And so this big first section, defining their mouse model to recapitulate DOCK8 deficiency, and then finding these TFH cells that are increased and that they have specific characteristics like IL-13 secretion, of course, the IL-4, but then they also express things like IL-5, transcripts GATA3, secrete less IL-21, and they're all associated with this hyper IgE state. So they're really trying to tell us that this is an important cell type that without the controls of this DOCK8 gene, and the point of this paper was not to help define what DOCK8, what that gene is, the regulation of that. I think it was just that we know when we knock it out, it can help expand this population because the regulation is gone. Now we can look further at, okay, what is this cell type doing in a wild type mouse? And so after they move on from their DOCK8 deficient mouse, and because we really need to know, can we find this cell type in a wild type mouse after it's been after allergic sensitization? And so what they did was they went on and said, okay, we're going to take wild type mice. So we're going to immunize both with the type 2 and type 1 model antigens. So they defined type 2 as, it's called Alternaria alternata, which is, it's a fungus, a plant fungus. And they took that protein, again, alone, it wouldn't induce as a robust response, but they paired it with the hapten ovalbumin conjugate, and they compared that then to LPS and then the ova MP conjugate. And so the type 2 model allergen was able to produce the high affinity IgE, just as in the DOCK8 mouse. But then when it was immunized, those mice were able to then generate a higher amount of also the high affinity IgE and anaphylaxis. So after that, they thought, well, okay, if we want to look at this cell type that's proliferating after this allergen response, are they transcriptionally distinct? And so they used single cell RNA sequencing, and they pulled out specifically these T follicular helper cells after immunization with the type 2 antigen. They extracted the RNA and then they sequence it. They process the data set that they get, that you get tons of transcripts, and you really are trying to then form different clusters with distinct transcriptional signatures. And so that's kind of where, Vincent, you had mentioned there's all these different types of TFH cell populations. They describe TFH2, TF regulatory, and then they're describing this T follicular helper 13 population. And they express particular markers, and they mention, again, this GATA3, IL-13, and these high levels are really distinguishing it. When you look at these plots in their paper, they're separated or distinct from the other populations within the T follicular helper cell subset. So they're further trying to define for us, this cell, is it distinct? Is it redundant? And so they move to flow cytometry. They do intercellular staining of IL-4 and IL-13 double positive, again, after they stimulate with the antigen. And so they were able to then further distinguish that these cells are also upregulating BCL6 and a couple other markers that are associated with T follicular helper cells. But then the transcripts are even found in unstimulated T follicular helper cells sorted from mice, but not when those mice are stimulated with LPS, which is that type 1 response. And so I think they're trying to show us here that IL-13 transcripts induced by the type 2, not the type 1 responses, is really what's defining the cell type, the cell subset. And that really, it's only during type 2 responses is this cell type induced in conjugation with the high affinity IgE responses. So they're able then to show in wild type mice that this cell type is induced, it's distinct. And so given this phenotype, they also want to know, is the location of these cells in the same place as one would expect? So is it in the same place as other T follicular helper cells? And they used something interesting. They used a reporter mouse that what they did was, it's called SMART13. It's a knock-in mouse. And so what they do is it expresses modified human CD4 from the mouse IL-13 locus. So basically, if IL-13 is expressed, then you would see CD4 expression concordant with IL-13 intercellular staining. So they show a picture here where the location of these T follicular or 13 cells is in a similar location in the germinal center and the T cell zone corresponding to TFH cells.
So this is, why do they have to do this? Why don't they just look for IL-13? They don't have an antibody or what?
Yeah, they should have an antibody. I wondered that too, because it's an involved process to try to
I have a thought on this.
Okay, I'm ready.
If you're going to try and stain a soluble cytokine in a tissue, it's going to kind of be everywhere. If you want to know which cells are making it, typically what we do is we engorge those cells. So you basically stop them from secreting anything so that you can stain it on the inside of the cell.
Yeah.
When you're in a whole animal, that's really hard to do. We can do it kind of with the populations of cells that are circulating in the blood. Sometimes you can pull out splenocytes or lymph node cells. I don't know how deeply the drug that you add to block secretion gets into the tissues in enough concentration to block secretion so that you can see the cytokine staining on the inside of the cell. So usually what we do is we'll treat cells ex vivo, so take the cells out, re-stimulate them in the presence of this blocker, and then see if they were making the cytokine. Or you can do it in vivo sometimes, but I think if you're doing tissue staining, it'd be really, really hard.
Okay.
It's not impossible, but it's hard. So I think using this surrogate, you can actually identify those cells that were making IL-13.
Because CD4 is a plasma membrane protein.
Right.
It's a plasma membrane protein. It's also not in a mouse because it's human.
Yeah. Well, they also say that the human CD4 was concordant with IL-13 intracellular staining. So they actually did both.
They did.
And so I don't know if they used the inhibitor or not in this experiment. They must have, right? But this is in mice.
They could have, yeah.
Anyway.
Right.
Okay, got it.
So up until this point, are we convinced that they were able to define this particular cell population in mice?
Mm-hmm. Sure.
Yeah, I buy. I want to know how many there are, though.
Not very many.
Yeah, I mean, not
in a
very
mouse,
many.
like million?
Yeah.
Yesterday, I was talking to a neurologist, and I said, "But is there significant pleocytosis in the cerebrospinal fluid?" And he said, "Well," and to a neurologist, that could be 10 cells.
Yeah, anything, something.
So do you think there are a million cells in the mouse, or 10 million, 100 million? No idea. They don't really say. So
I don't have a good reference.
Yeah, I don't either, but I don't think it's going to be any more than, if T follicular helper cells are, let's say like 2 to 3%
right.
of the T cells. And then within that subset, we're looking at one that's only upregulated during allergic sensitization or during this very extreme, the DOC8 mouse model is a really extreme hyper IgE. I don't even think, they showed the same thing when they gave wild type mice an allergen, but I don't think it's as an extreme of a phenotype. So I would imagine it's a relatively low number.
All right. So if you have 100 million cells in a spleen, I just looked this up, about 20 to 30% of them are T cells.
Mm-hmm.
And if you look at CD4 T cells, about, let's say roughly, it says 13 to 20%. So let's say about 15% are CD4 T cells. So if 15% of the total CD4 cells in a spleen or in a lymph node, I'm just, making a correlation here because I don't know the exact numbers, are CD4 cells. And then these are what percent of CD4 cells? Is there any flow on here that can tell us that? I don't remember.
They're panels in the supplementary, but I'm not
a couple percent.
Yeah,
it's the total
from my
CD4s.
memory, it's only 1 or 2%.
So it's you're talking probably less than 1% of all the splenocytes. This is a lymph node. I can't remember how many cells you get from a lymph node, but you get about 50 to 100 million cells from a spleen, and so less than 1% of them are going to be these cells.
Now. Yeah,
right.
So just the TFH cells were at 30,000.
And if you talk about there's a bunch of lymph nodes in the body, it's probably 1 million. That's a rough guess. Somebody can correct me, but that's my on the back of a napkin calculation.
But it is fascinating. As they work through these models and they're knocking out TFH cells, they're knocking out, we'll do it in a little bit, but particularly the ones that produce IL-13. They see a pretty robust increase in IgE that this one very rare cell type is kind of driving the IL-13 secretion, the hyper IgE, the anaphylaxis. So I think it's so far pretty convincing that this cell type is doing something pretty important.
Mm-hmm. Just because you have an abundant cell type doesn't mean it does everything. And just because you have a rare cell type doesn't mean it's not critically important.
Mm-hmm. Right. And so why it also leads to the point of why this cell type is probably highly regulated by DOC8 being one gene, but you would not want this cell type to overstimulate or hyperstimulate IgE because obviously it packs a pretty powerful punch for only being a very small percentage of cells. So the next thing they wanted to look at was they showed it in these DOC8 deficient mice, they showed it in wild type mice, but do they see it in humans? And so what they were able to do is to look at human cohorts that were sensitized to either dust mite, peanut allergy, and then aeroallergens, which are things like seasonal plants that cause hay fever. And so what they found was that compared to non-sensitized individuals to any of these different allergens, that the sensitized individuals had a higher frequency of these TFH13 cells defined by the things I mentioned before. So defined by the transcripts, defined by the secretion of IL-13. And so I thought that, they are seeing an upregulation of this particular cell type, although I think it could be a dysregulation in the immune system in general, and that the increase in that cell type is due to the increase in another cell type. So what I think they would need to do in terms of human samples is maybe do some type of stimulation and see if you can increase the frequency of those cells, or if you could, it's, I think, difficult. And they're using pediatric patients. So, to get some type of allergen study, I'm sure would be challenging, but it's not necessarily a direct correlation, but they do find them in human patients.
So there is one model system where if you have an allergy, they allow you to do human studies.
Is that
celiac disease?
Oh, okay.
But that's not mediated by the same immune effector mechanisms, so that doesn't help us.
Right.
But you are allowed to do that because if they eat gluten, it doesn't make them deathly ill, and they can stop eating it and their symptoms go away. So they do actually look at that in real time in humans. But that one of the other things that's really interesting is, yes, they're using pediatric patients, but the other thing they're doing is with humans, you sample blood, right?
Right.
So we started off by saying these are follicular cells in the follicles of germinal centers and lymph nodes, right? So you're getting a small sampling of perhaps what's going on in a human, but it's not exactly what's going on. Because in a mouse, we can euthanize them, we can take their lymph nodes, we can grind them up, or we can cut them up and we can see what's going on. We can't do that in human.
Right. Yeah. So that would be, and I guess, the other thing, and I didn't really think the anaphylaxis induced by, they described that ear test, but wouldn't the mouse die? They described that some of these mice actually get quite old and they just have this hyper IgE, but I guess it's not really recapitulating the anaphylaxis induced death. It's not really.
So they did, they're using a percutaneous model. And so they put a dye in the blood of the mice, and then they look for vascular permeability in a local tissue, and the ear is really easy to do that because it's so thin, and you can measure that. You can do systemic anaphylaxis in mice, but I think it's just a different setup. It's not exactly the same. The PCA is a little bit easier to do.
Right. Okay. So the next thing they did was they wanted to look at helminth infections. We mentioned that helminth is a type 2 immune response. And is it the same? So previously, we know that these T follicular helper cells showed IL-4, but not IL-5 and IL-13. So what does a helminth infection do in terms of this cell type? And so they infected mice with Nipostrongylus brasiliensis. Vincent, do you guys talk about that on your Twitter? I've heard it on your Twitter.
We have a guy here who works on it as a type 2 antigen. Yeah, totally. Okay. It's a mouse helminth. So people who use mice do that all the time. Yep.
Right. And so what they found was that when you co-immunize with this helminth, with that hapten carrier protein conjugate, the MP-OVA, it does elicit TFH cells, but it does not elicit TFH13 cells. And GATA3 was not upregulated in the TFH cells. So there was no IL-13 production. So they're absent, but you do have IL-4 positive, again, not IL-13, Th2 cells present. So you have it. There was a strong IgE response, but but little of it was the high affinity. And so it really induced more of this low affinity IgE, and was the association with the TFH13 cells from previous experiments really is the Th2 induced response. So the alternaria alternata immunization and not the helminth. So it seems that helminth responses may be responsible for a different type of type 2 response than these T follicular 13 cells, which I thought was interesting that, this is really the divergence. And when we're asking the question whether this cell type is specific or if it is a transitional cell from Th2 cells, because they do share similar properties, that in terms of helminth, the responses do diverge and they may be distinct.
And I think it's really interesting that, this is one way in which you can look at a fundamentally different immune response between two things that induce the same type of immunity. I don't know if that's making any sense, but we talk about the hygiene hypothesis. We come back to that again. If you have helminths, they're driving this low affinity IgE.
Right.
And perhaps if you are continuously inducing that type of response, you are promoting fewer TFH13s, so you have less potential to drive anaphylactic responses. Right. Whereas if we don't have those helminths around anymore, maybe there's just a higher propensity to drive these TFH13s. And so we now are developing allergies that we didn't develop before.
Right. Yeah, I think a lot of what you're saying, it really lies in our ability to kind of define the mechanisms of why a helminth infection are different from allergies in terms of the cell type, because it is a pretty robust cell type. And so, that was the helminth part. And then they kind of end on using a mouse model where they were able to abrogate only IL-13 producing TFH cells. So it's kind of like leading up to this. Okay, before they deleted just TFH cells, the BCL6 knockout mice, but now they're actually removing IL-13 expressing TFH cells. And so they did that, they found that there's a loss of IgE germinal center B cells. There's a decrease in IL-13. But they did still find the same amount of total IgE, which was interesting. So even though they didn't find this high affinity IgE when they knocked down those TFH13 cells, there was still total. So is there this compensatory mechanism for IL-4 Th2 cells or T follicular helper 2 cells that may be sufficient to promote the total IgE, but then the other TFH subtype, the TFH13, that's the high affinity. And so by being able to knock out just the IL-13 producing, they're able to kind of define more specifically within this TFH population, which one is more responsible for the high affinity.
They don't do the NIPO experiment, though. You would expect that to be normal, right? And that knockout.
Yes, that would have been nice to see. Yeah, that you would expect that to be normal. And that would have been nice because we could have better defined. I guess they could dive into that helminth infection more and delete maybe some TH2s, but maybe the helminth part was just to see the difference. And then that's like a future aim for next paper is looking at the cell type. And so the last thing that they did was, they did these mixed bone chimera mice, which mixed bone chimeras are mice that are irradiated. It kills their bone marrow. And then all the bone marrow derived cells it's from another mouse. So you just give that irradiated mouse bone marrow from another mouse. And they gave the irradiated mice bone marrow from the IL-13, the TFH13 knockout mice as donors. And those mice did not produce IL-13, but still made the IL-4 after immunization with the type 2 allergen. Again, comparable titers of total IgE, and it was low affinity. So they were just able to recapitulate what they found in a mouse that did not have bone marrow. And so I think what all of this shows us in the end is that I'm pretty convinced that this is in fact a new cell type. These T follicular helper cells that produce IL-13 is required for the induction of IgE that does induce anaphylaxis in mice. I think when thinking about targeting for treatments, this could be a cell type that you would target, but it goes back to, I think they brought it up, what would be, is this a transitional cell type between Th2 or not? I think they have to kind of further help define that for us. And whether you're going to induce high affinity, there's checks and balances on the cell type. So in what circumstances would these be induced? I think they have to further look into that. But yeah, what do you guys think? Do you think that you are convinced that this cell type exists and is important?
Yeah.
Yeah, it looks good to me.
Yeah.
I would like you to review what they say about how this is made by switching, 2 different switchings to make the 2 different affinities. Do you know that? Because I think that's pretty cool for people to hear.
Yeah. So you mean in terms of what they propose?
Yeah. We propose a two-tiered model of IgE induction. Yeah. Can you summarize that?
Yeah. So what they're proposing is that, so we described that these TFH cells, there's different subsets. And so the TFH2, not the TFH13s, maybe instruct the switching to IgE plasma cells in an IL-4 dependent manner that results in the low affinity IgE. But these TFH2 cells, which they've shown in this paper, they don't express GATA3, they don't secrete IL-13. But in contrast, allergens that induce the GATA3 positive, IL-13 positive TFH cells, they may instruct the switching of IgG1B cells. So if you remember back to the beginning when I first started talking about this, what that DOC8 model showed us was that there, even though it was a type 1 driven immunization, but they had an increased amount of hyper IgE and a decreased amount of high affinity IgG1. And so that was suggesting that potentially that there's this switching from IgG1 to IgE, which would then enhance, the anaphylaxis part of the response to that mouse. Because
it's sequential switching, right? That drives the high affinity IgE from IgG1.
Yeah.
And that's, they think, is somehow instructed by the TFH13 cells.
Mm-hmm.
Right, which is interesting because IgG1 would have already had signals to drive it to IgG1, and then the sequential, like you said, the sequential switching compared to if it's just an IgM1 positive, then being induced to IgE, that would be that low affinity. So
that's the key. That's the IgM to IgE is the low affinity because presumably there's no TFH13 cells around to make it do the other thing. I think that's cool theory.
It's very cool, yeah. And, I think that further, when they look at the loss of IL-13 and it doesn't impair IgE responses, particularly where low affinity IgE, in helminth infections, when they did that experiment, it's consistent with that two-tiered model of IgE.
Yeah. So, Steph, they say now we have molecular targets for therapeutic treatment of allergy. So what would the targets be? These TFH13 cells, some IL-13. What do you think?
Well, I think you would have to inhibit the generation of high affinity IgE. It's as a way to block the severe allergy responses since they express GATA3. I guess you could use like a GATA3 enzyme that would remove GATA3, but then you might have issues because GATA3 is also Th2.
You need a small drug, right? A small molecule, and I guess you could develop
antibodies.
one. You could make antibodies, I guess. Yeah, something to inhibit
IL-13.
13 would be it, right?
Yeah, so they have it. So that's why I was looking up. That's why I was quiet.
They already have it.
Yeah. So in 2016, MedImmune and AstraZeneca were developing traqualinumab for asthma, which is anti-IL-13. While clinical development for moderate to severe ulcerative colitis and idiopathic pulmonary fibrosis have been discontinued. But in July, AstraZeneca licensed tralokinumab to Elio Pharma for skin diseases. But I don't see anything about allergic diseases.
Well, I would imagine that would be of high interest after this paper came out.
Yeah, for sure.
Yeah, I'm sure they're looking into it.
Yep.
I think it's going to be hard though, because I think you would only have an indication for this in individuals, one, that are not too susceptible to, I guess you don't need 13 for the low affinity ones that are involved in the parasites.
Right.
So that doesn't matter.
So
but I think you'd have to have like pretty, familial inherited, connection for developing life-threatening anaphylactic food allergies, for example, or something like that, because you'd have to start treating with anti-IL-13 really, really early and continue for a long time in order to prevent any generation of IgE, because you don't know when the exposure will happen, right?
Right, exactly.
So I think there's challenges to this. I mean, on first thought, it's like, oh yeah, you just block IL-13, and then you're like, wait a minute, maybe that won't work.
So they say here, IL-13 remains one of the most replicated genetic associations with elevated IgE food allergy. blah, blah, blah. So that's why they made those monoclonals.
Right. That's right.
It says it will be interesting to see whether monotherapies targeting IL-13 in patients affect more than tissue-restricted pathology. So, I don't know if a monoclonal is the best, but you could take a small molecule, a drug, all the time, because that's what people do in the spring. They take that.
That's true.
Antihistamines.
Antihistamines. What's the name of those drugs that my daughter takes?
Loratadine is a generic version, but it's Allegra.
And what's the other one?
Begins with a Z.
Yeah,
I don't remember.
Okay. And they take them all.
They're
the
all
time,
available over the counter now.
Yeah.
They weren't a few years ago.
Oh, and then this is what you mentioned.
Zyrtec,
right?
Zyrtec, yeah.
Benadryl.
GATA3 deoxyribozyme to inhibit GATA3 activity. It's been used in asthmatics, and it reduced the eosinophilia. What's a deoxyribozyme? A GATA3 deoxyribozyme. Does anybody know what that is? Deoxyribozyme. DNA enzymes. DNA oligonucleotides that have that can be catalytic. Well, I guess you direct it against the
GATA3. Yeah. But you're right. You would have to be early, and if you're taking it for a long time and you're knocking down GATA3, that's also a Th2 factor. So that would be problematic.
That'd be a problem. Yeah.
Right.
So there's a paper here that they say describing it. Blood eosinophils predict therapeutic effects of a GATA3 specific DNA zyme in asthma patients. Huh.
That's cool.
Amazing stuff. Yeah, it's really cool.
Very cool. So I think it's exciting when we find new cell subtypes in immunology. It's exciting to kind of follow then that cell type as it develops, because surely tons of people are now going to be looking at this particular TFH13 cell and what it does in a variety of different types of circumstances. So I thought it was a good paper. I enjoyed it. It wasn't, I followed a story. It wasn't particularly challenging to read, which, is pretty good for an immune paper. So yeah, I liked it.
Yeah.
No, I don't know. It's challenging for me because these graphs are too small. I can't see them.
Yeah, I know. It's
they
terrible.
put 20 plots on a page. It's just ridiculous.
I know.
They do.
But like figure 6 and 7 were pretty big. That's pretty nice. Maybe a couple.
That's because you're 20 years old, man.
No, not 20.
Then the supplementaries are also it's hard. Within the era of online, I don't know why things have to be so small, because they want to fit it into this, page format still, because they want to make a print. So we have to get beyond that. I think it was very good. I appreciate that you picked it, because I'm not sure I would have looked at it otherwise, but it's really cool. And it makes a lot of sense. Yeah.
Yeah. I know it fixed something in my head that has been bothering me. You teach these things, and you need IL-4 for immunoglobulin class switch, and IL-4 is a Th2 cytokine and helps drive IgE. And everybody always says, well, but then if you have IL-4 and you need that for any isotype class switch, how do the cells know to switch to IgE? And I said, well, those are things that immunologists study. It's too detailed for me to go into right now. And then I'm like, wait a minute, I don't know that. Now I do.
Now you do. Now you can say
now I do.
TFH13.
So but it's good that I didn't know it, because I just didn't know it and it was known. It was because it wasn't known. So
Right.
All right. Let's do a couple of emails here. Is that okay?
Yeah.
Sure.
First one's from Ryan, who sends a link to a article on a new HIV vaccine study. And he says it's promising, but we have to wait and see how the FDA responds to the results. So he sent a link to a CNBC article which says J&J to test HIV vaccine in the U.S. and Europe. But the article says nothing. So I looked it up. Mm-hmm. And there are two trials that are going on here that are interesting. One is a trial. This is run by NIH. It's a HIV vaccine efficacy trial in the Americas and Europe, and this is involving a mosaic antigen made up of different HIV types. and the regimen, so it's being delivered in an adenovirus vector. They're going to get 4 doses of an adenovirus vector encoding this mosaic antigen, and then they're going to get purified envelope protein, which is also going to be mosaic. So that's interesting. And then the other one is a trial in Africa, in South Africa, which doesn't often happen. Mm-hmm. And this is using a vaccine that was used in the famous Thai trial called RV144, which was 30% effective, which is almost placebo effect. But everybody was happy because it was the only time that any effect was seen. And this is a prime boost strategy where they gave canary pox vectored glycoprotein followed by purified glycoprotein. So they're going to do that in South Africa. So there are lots of big HIV. It is also sponsored by NIH. There's a lot of big HIV trials going on. I don't know. It's I think you have to do it, but aside from that 130% efficacy, nothing else has worked.
I know. Do you think we'll ever have a vaccine that'll work for HIV?
I asked. I did a podcast, as I said, in Denmark, and one of the people was an HIV person who's trying to do immunotherapy, right, to try and get your immune response to clear. And I asked him that. I said, "What do you think?" He said, "Maybe one day, but it doesn't look good right now." So I think we don't have the right.
Hedging the bets.
I just think we don't know what to induce for protection.
I think it's hard. I think those broadly neutralizing antibodies, I think there's a reason our body does not produce them at any high rates. I know that was exciting to find those, the people that were high controllers but broadly neutralizing antibodies are an antibody. I don't think that they can be induced at high numbers in many people because they're probably highly reactive to other proteins in your body. And, the confirmation of HIV and how much it changes, it's just very challenging.
Yeah. Cindy, can you take the next one?
I sure can. So Scott writes, my wife has been treated for non-Hodgkin's lymphoma for the past 3 or 4 years. She did outpatient treatment first, achieved short-term remission. Then they did stem cell transplant, and that worked, quote unquote, for a while. Recently, she was selected to participate in a CAR T study. She is again in remission, 7 months so far. I very much appreciate your ability to make this very complex topic slightly more understandable by the layman. Again, many thanks. Well, first of all, best wishes for your wife. This is amazing. Definitely. Third time is the charm. CAR T is going to work. I just know it. I have my hope. Yeah, it's amazing. It's amazing. All of those things, all of the treatments she's done probably have arisen from incredible science and research that went into that. And if we've done anything to help explain that so that you and your wife understand what's going on in these therapies, you're welcome. That's amazing. That's why we're here. Thank you.
Yes. And I'm glad that he found, or she found, Scott, yes, that it was understandable. I mean, often I worry that I'm not able to make it understandable. So it's great. But also, Cindy, you did the CAR T cell episode, and I listened to that, and it was excellent. So I'm sure that helped a lot.
Steph, can you take the next one?
Sure. Diraj writes, hi, Cindy, Steph, and Vincent. I was listening to Immune 17 on CpG stimulating macrophage metabolism to potentiate anti-tumor activity a couple of days ago on my commute home from work. And I immediately thought of this episode when I came across this paper when I was perusing PubMed for new immunology studies. Here's the title: Programmable Bacteria Induce Durable Tumor Regression and Systemic Anti-Tumor Immunity. This paper is so cool. They program non-pathogenic E. coli to lice in the tumor microenvironment and deliver an anti-CD47 single domain antibody, nanobody, against tumors that express CD47. They ended up seeing evidence of increased tumor infiltrating T cells, higher numbers of class 2 expressing macrophages, tumor regression, and reduced incidence of metastasis. homeostasis in multiple syngeneic mouse models. I thought this added a nice adaptive immunity layer to the paper discussed in Immune 17, and it seemed like something that would be interesting for you all and other listeners. The paper was published in Nature Medicine on July 3rd, 2019 by a group at Columbia. They list the authors there. I think the last author's last name is Danino, and hope you all find this to be as cool as I did.
That sounds awesome.
Very cool. Yeah, very cool.
So we did this on Twim because
you did?
Nick Arpaia and Tal Danino are my colleagues here. So I said, come into the office. It's an amazing study. It's Twim 204.
Oh, awesome. There you go.
Very cool.
So you didn't get the immune perspective, but I'm sure they did a great job.
Yeah.
It's very, so Nicholas is a immunologist.
Yeah.
He trained with some good immunologists. And Tal is a guy who he's a bacterial geneticist. He worked with Jeff Hasty, who is the guy who devised this idea of using quorum sensing to make bacteria undergo cycles of growth and lysis. So these E. coli are programmed to grow lice. Okay. There's always a few left, and then they grow and then they lyse. So they are perfect for delivering things. So they put this nanobody against CD47. Amazing thing is when you inject E. coli into mice, they're cleared unless there's a tumor, in which case they go to the tumor. Only to the tumor.
Whoa.
They only go to the tumor. And I'm thinking, oh my gosh, it should kill them, but no. And then in the tumor, they grow lice, and they deliver an antibody to CD47, which they said CD47 is a molecule on the surface of cells that says, don't eat me.
Yep.
And so you block it and the cells are eaten by phagocytes and so forth. It's really brilliant. So go listen to them talk about it.
Awesome.
It's just a great combination of, microbiology and immunology, right? It's great.
Love it.
And the cool thing is, one last comment, you can give the bacteria intravenously, right? Works great because then you can hit all the tumors.
Wow.
But they did an experiment where they injected the E. coli they put a tumor on both sides of the mouse, right? These are implanted human tumors into mice. They inject the E. coli into one tumor, it regresses, so does the other one without E. coli going to it. So they're activating general immunity against the tumor.
Yep. So there's this idea that you can break tolerance to tumors if you have a strong infectious disease. So this was coli. Mm-hmm. 100 years ago, tried to treat people with tumors by giving them severe bacterial infections, and sometimes it worked.
Yeah, that's right. They talk about how that began. They noticed that people who had these infections had their tumors regress. So it's progressing into a very interesting therapy. Yeah.
Yeah.
All right. One more is from Renee, who writes, hello, an educational YouTuber called Kurzgesagt. You probably know of him, released a video about the complement system. You should check it out. And yes, Kurzgesagt is a great YouTube channel. I think that means how it works in German.
Oh, is that what it is? Okay.
It's really good. And really nice animations.
Yeah, it really was. I went and watched it. And it's factually pretty darn good too.
Yep.
Yeah, it is.
Good. I'm a 25-year-old female, graduated from University of Puget Sound with a degree in chemistry and math, currently research assistant in Seattle, working at the Infectious Disease Research Institute. My group works on screening unknown research compounds for potential treatment against tuberculosis. I hope to go to grad school eventually for microbiology or something involving both micro and chemistry. I listen to all your podcasts and appreciate all the work you do. Thank you, Renee. Renee, do you know Mark Owen Martin? He's at Puget Sound. You must know him if you love microbes. All right. Some picks of the week.
Yes. Okay. I have to tell you that my picks of the week. So Vincent, your pick of the week that's coming up, Nathan had sent me that.
Oh, I'm sorry. And you were going to pick it?
I was going to pick it. And then I thought, we'll talk about the Lasker Award because it has to do with immunology. That is Cindy's pick. So I think I'm just going to say that your picks are excellent. And I'm just going to pass it on to you. You guys can explain your picks.
Sorry.
No, that's fine. No worries.
It's a competition. Vincent puts up our document that we put online. I'm like, I got to put my idea for the pick of the week so it doesn't get stolen. So I beat you to it.
Yeah.
So you want me to go?
Yeah, go ahead.
Okay, I'm really excited about this. So the Lasker Award is a huge award, and it was just given to two immunologists Max Cooper and Jacques Miller for their amazing seminal discoveries in the identification of distinct classes of lymphocytes. So everything we talked about today is talking about a specific subtype of T cells. Well, originally it was thought T cells and B cells were the same thing, or there was one thing and nobody really knew what was going on. From the press release, it says this pioneering work that these two individuals did has fueled a tremendous number of advances in basic and medical science, several of which have received previous recognition by Lasker Awards and Nobel Prizes. They include monoclonal antibodies, generation of antibody diversity, MHC restriction, antigen processing and presentation by dendritic cells, checkpoint inhibition for cancer therapy. None of that would have happened without these discoveries. And so Jacques Miller was studying a cancer-causing virus, and it turns out that they were trying to chop out the thymus and see what it was useful for, and they discovered it was important for antiviral immunity and also for skin transplant rejection. So he found out there was one subtype of cells that did this, and the thymus was really important for that. And he wasn't believed at first. It took a while. But Max Cooper studied something called Bruton's agammaglobulinemia, which is kind of the opposite of what you were talking today with IgE syndrome. So this is a lack of antibodies. And he used a chicken model because there was someone named Glick who published that the removal of the bursa fabricius in little baby chickens that just hatched could lead to loss of antibodies later. And Cooper published in 1965 and 66 that there were these two distinct lineages of immune cells arising from the two different organs, the thymus and the bursa. And so this is just understood in immunology. Now, no one questions this, but those discoveries aren't really that old in the context of biology.
Right.
1960s. And so in 74, he actually found that the B cell precursors in mice were in the bone marrow, which we know in mouse and human and all these other vertebrates, besides chickens, have their B cells are in the bursa because we don't have bursas. Bursas are in the butts of chickens. So anyway, the Lasker, you guys can probably add to this, is really often considered the precursor for the Nobel. And I actually hope it happens this year because these two individuals are really old. Max Cooper's 86. And I think Jacques Miller is older than that. And so they incredibly deserve this award. It's amazing. And Max Cooper actually still runs a lab. He's 86 and still runs a lab. Anyway, I also thought it was kind of amusing before I go on and let Vincent talk about his, is that you got to love the creativity of some of the people who write these lay press articles, because I was looking up some, things about this, and somebody wrote a title of an article, How Studying Chicken Butts Cracked the Inner Workings of Our Immune System.
So corny.
Yep.
And I thought, wow, those poor guys. That sums up their entire life's work. That's deserving of a Lasker Award and a Nobel Prize. They studied chicken butts.
Love it.
So my pick was sent to us by Steph and I have a friend down there in the Triangle area, Nathan, right? Who sent this video from Ellie, who's a senior in high school in Connecticut. She's a summer intern with Akiko Iwasaki, and she is a semifinalist in what's called the International Breakthrough Junior Challenge Science Video Contest, which inspires students to think creatively, submit videos up to 3 minutes in length that bring to life a concept or theory in the life sciences. So out of thousands of submissions, Ellie's video on cancer immunotherapy has been evaluated as one of the top 30. So you should awesome. Put the link in here, the Facebook link, and you can like it or share it. Each like and share gets her a vote. So check that out.
Let's do it.
Help her out.
Definitely.
So I will. The deadline is September 20th. So I will put this immune episode up tomorrow. I'll push it out quickly so that people can see this and help her out. So that'd be cool. So Steph shared both of those picks with us. All right, microbe.tv/immune, immune at microbe.tv for questions and comments. And if you really like what we do, you could support us financially because we all do this on our own dimes. Microbe.tv/contribute. We have Patreon or PayPal mechanisms where you could give us as little as $1 a month, not even a cup of coffee. Send it this way and we'll continue to make great science for you. Cindy Leifer's at Cornell University. On Twitter, Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langel's at Duke. Stephanie Langel on Twitter. Thanks, Steph.
Yeah, thanks. This is fun.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.